Okay, welcome back to part 2 of chapter 10, talking about muscular contraction and how does it all work. So now that we understand how the sliding filament theory contributes to movement of a muscle contraction, we are ready to go through all the biochemical mechanisms step by step, starting from an electrical signal that's coming from a neuron, and how do we go from there to a muscle contraction. Now, in order for a muscle fiber contraction to occur, it must first be stimulated by what's called a motor neuron, okay? We'll talk about different types of neurons when we get to the nervous system chapter, but motor neuron are the ones that are specifically innervating or stimulating muscle fibers. So that's why it's called motor neuron, because it is for movement, okay? And remember from chapter 4 that we glossed over the basic structure of a neuron, where there is a cell body. This is the cell body right here. So this whole thing illustrated by this picture right here is the neuron. And this right here is the cell body that we talked about in chapter 4. These little branches coming off are the dendrites, okay? That's the cell body. And dendrites is for receiving signal, because, it doesn't go from one neuron and then from the brain, one neuron, and then there's your muscle. It's not that simple. Usually it's your brain as a central nervous system. It goes through your peripheral nerves, okay? Peripheral nerves, sometimes it's one neuron, sometimes it can be multiple neurons. So there can be another neuron right here with another cell body, okay, and then the axon like this, and connects to these dendrites and sends signal from, for example, your brain towards this neuron. This neuron tells this neuron to do something, so this neuron receives that signal in the dendrite. Okay, let me change color again. Okay, so that's the brain. So the neuron then sends the electrical signal to this neuron. This neuron receives those signal as dendrites, and then those signal then gets propagated towards your muscle fiber. Okay, and this part right here is what we're gonna look at, right here, from neuron to neuron stimulation. What we will be talking about in chapter 12. Okay, so that's the hard part with chapter 12, is that action potential from neuron to neuron. Okay, how does it get propagated? All right, so now there are myelin sheath. Okay, so these little yellow, I like to call them as, those not ho-hos. What is that again? Your I forgot what's that sweet's called, but it almost went out of, it was almost going to be stopped production. Anyway, Twinkies, okay, Twinkies. It looks like little Twinkies wrapping around your neuron, axons of the neuron, okay? So those yellow Twinkie looking thing is what's called myelin sheath, okay? And myelin sheath is what wraps around the axon to allow the neural conduction of that electrical signal, or an action potential, or what we call electronic stimulation, a lot faster. We'll talk about what is the purpose of having myelin sheath, and why some neurons don't have myelin sheath and some neurons do. Okay, not all of them have these Twinkies around them, but only some, okay? But whenever we have them, it is for the fast conduction of the electrical signal, okay? So this, again, will be a concept we'll revisit in chapter 12. But we're not focusing, neuron is not our main focus in this chapter. The main focus is what's happening right here, okay? Between the neuron and the muscle, okay? So at the end of the axon, so this is myelin sheath, wraps around the axon. The axon is continuous going down, and then you have these nubby hand looking thing. I always like to call them a hand. It kind of looks like a hand with a lot of fingers or something. And that hand with fingers kind of sits on top of the muscle fiber just like that. Okay, and that's what we call the connection between the neuron and the muscle. Okay, and we call that the neuromuscular junction. That's what we mean by neuromuscular junction. Okay, so it connects, that neuron is able to innervate that muscle fiber via this connection between the neuron and the muscle within this neuromuscular junction. Okay, There's only one NMJ. NMJ is short for neuromuscular junction per muscle fiber. Okay, you can't have two neurons innervating the same skeletal muscle fiber, or else, the fiber will be really confused as to which neuron signal should it receive commands from. You can only receive from one neuron, so then they all contract together. If two neurons are sending electrical signal at different time, then you're in trouble. The muscle fiber is like, okay, should I listen to neuron 1 or neuron 2? They're coming in with commands at different times, so I'm confused. Okay, so we don't want that to happen to the muscle fiber. So that's why you only have one neuron per muscle fiber. Okay, now one neuron can actually connect to multiple muscle fiber, and we'll talk about that towards the second part, towards the end of the chapter. Okay, now the other thing I want to mention here is that the axon terminals are not exactly connected to the motor end plate physically. Okay, what do I mean by motor end plate? So motor end plate are these indentation you're seeing in the sarcolemma. Okay, remember, sarcolemma is the plasma membrane of the muscle fiber itself. Okay, so the sarcolemma forms these curvatures in this specific spot where the neuron is attaching to. And by attaching, I don't mean physical connection, but it's just kind of sitting there with a tiny little gap between the neuron and the muscle. So this curvature area is what we call motor end plate. Okay, think of it as on the muscle side for the motor, and a plate, okay, a little plate for the neuron axon terminals to sit on. Okay, so this is those nubs that you're seeing right there, and one of those is called axon terminal, and the axon terminal is what sits on top of the muscle fiber, not the axon or the myelin sheath or the cell body of the neuron, just the axon terminal that's sitting in the motor end plate. Okay, and now when I'm, like I said, what I mean by sitting in there is not a physical connection. It's separated by a very tiny gap called synaptic cleft. Okay, or some people call it synaptic gap, but usually it's named by synaptic cleft. And that space in between is what allows chemical signals to go from the neuron onto the muscle fiber in order to stimulate its contraction. Okay? All right. So let's begin by talking about how the neuron transfer the signal to contract, cause the muscle fibers to contract. How do your neuron and muscle fiber communicate? Okay, so let me start with a little analogous story, because people always find this mechanism very, very confusing. So I'm gonna start by talking about a story that you're familiar with. Like I was saying, with anatomy and physiology, you have to relate it to something that once you get it, you never forget what it is again. Okay, so now I'm gonna try to explain it in terms of something you're familiar with. Okay, so this whole process that's happening in the NMJ is kind of analogous to your boss needing to give you a task to do your job. Okay, but he as a CEO of the company, usually they don't directly call you as an employee with that task. Okay, there's a whole chain of command that your boss has to go through, so he doesn't need to communicate to 10,000s of his employees. Okay, so he, as a CEO, he will probably email your senior manager, and that's something, that something needs to get done. Okay, so he emails your senior manager, hey, you need so-and-so to get this done. Okay, well, which then your senior manager will email your direct supervisor about the task that you have to do, which will then your direct supervisor will let you know that you need to do blah, 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 because the CEO says he wants you to do this. Okay, so this whole process at the NMJ is kind of like that. The brain, let's say, is the CEO of your company, which is your body. Okay, then you have all these senior managers working for him, like these motor neurons. Okay, so the one senior manager that is responsible for your chain of command, that particular muscle fiber we need to relay the signal to, will relay the message and signal to your direct supervisor. Okay, your direct supervisors are the axon terminal. You as a little muscle fiber sitting there waiting for tasks to be done. Okay, you're waiting for that message to come from your supervisor. Your direct supervisor is the axon terminal. Okay, axon terminal then will release an email to you in form of neurotransmitter, okay, to tell you what to do. Okay, so that email you receive from your direct supervisor is just like how the axon terminal release neurotransmitter to tell the muscle fiber what to do. Okay, and then the muscle fiber will contract and does its job, just like how you received your email from your direct supervisor and you do what you need to do and turn it in. Okay, so that's kind of like that whole story in a nutshell, what's going on here at the NMJ. CEO, the brain of your body, telling you what needs to happen. But, instead of emails and phone calls, your nervous system and muscular system communicate by electrical signal and neurotransmitters. Okay? Very similar story, but it's just in different formats. Okay? So now, what do you do as a muscle fiber that needs to complete the task? Here it is step by step, okay? So step 1, first, your direct supervisor, which is the axon terminal right here, okay, is now ready to have all the instructions and ready for you to send it over to you as a muscle fiber, okay? Now they're packaged in little vesicles called synaptic vesicle right here. Let me change it back to red, okay? So this is the synaptic vesicle. And then they are, what's in the synaptic vesicle is your neurotransmitter, okay? These neurotransmitters are specifically called acetylcholine. There's many neurotransmitter in your central nervous system, okay? But the ones that specifically for activating muscle fiber contraction is called acetylcholine, okay? Or abbreviated as ACH. And they're all packaged in the synaptic vesicle ready to be released. But it needs to wait for that right signal at the right timing in order for that to happen. Now, when the order comes in from the senior manager arrive, just like how that electrical signal, that's what this lightning arrow thing is representing, that action potential as form of electrical signal arrives from the neuron, all the way from the dendrites through the cell body, toward the axon, all the way down from the, that's what I was talking about. Dendrites, cell body, axon, all the way down to your axon terminal, arriving here at the axon terminal. And when the signal arrived, it will stimulate the release of these neurotransmitter, which are the little red balls here, called ACH. Okay, and the ACH will go into the synaptic cleft. Okay, so this right here is the synaptic cleft. Okay, just like how it's referring to. That's what this arrow is referring to, is that synaptic cleft between the axon terminal and the motor end plate. Okay, all right, so what is happening at step 3? And that's what happens, okay? When the action potential reach the neuron's axon terminal, it will trigger exocytosis of the acetylcholine into that synaptic cleft space, okay? So that's those red dots moving into the empty space. We'll talk about what those moon-shaped yellow things are at the last step, okay? Now, at the 4th step, the acetylcholine will diffuse across from your axon terminal to the synaptic cleft. And in the synaptic cleft, there are these purple things you're seeing on the motor end plate side, okay? Those purple things there are called sodium channels, okay? We'll actually distinguish it from electronically activated sodium channel. These are actually what we call chemical activated sodium channels. We'll talk about those again in chapter 12. That's the nitty gritty part about all these different channels. But for now, just know that these are the sodium channels on the motor end plate side on the sarcolemma of the muscle fiber. Okay, so once acetylcholine binds, okay, the acetylcholine travels in the synaptic cleft, binds to a particular sodium channel, which has a little spot here for the acetylcholine to bind to, it activates and opens those sodium channels. Once sodium channels open, then sodium ions is going to rush through, okay? And it is this sodium ion rushing through that's what's going to cause and activate and stimulate an action potential, okay? So that stimulation of action potential is then going to be regulated or propagated towards the rest of the entire sarcolemma for the entire muscle fiber. Okay? So let me say that again. The rush of this positively charged sodium ions going into the sarcolemma is what will then initiate another round of action potential to be started on the muscle fiber side, okay? Because the action potential was on the neuron side. Now we need it to happen on the muscle fiber side. And that's what's going to trigger the action potential on the muscle fiber side is by acetylcholine attaching to the sodium ion channel. Sodium ion channels gets activated, allowing rush in of sodium ions, which then activates the action potential on the muscle fiber side. And then we will continue on with the journey, which we will talk about next, what happens after that. So in a sense, this ACH or acetylcholine here is what transfers the need for an electronic action potential to travel from the neuron to the muscle fiber. Okay? And I will talk about that after I talk about this. Okay, so the action potential generated at the motor end plate. This is the motor end plate right here, where you have the axon terminals sitting on your muscle fiber. So that action potential is now going to go travel along the entire length of that whole cylindrical length of the muscle fiber. It's going to travel along the whole sarcolemma, sweeps across the entire rest of the muscle fiber. So all the sarcomeres will know to contract at the same time. Now we are spending some good minutes talking about through this whole process step by step. But in reality, all of this happens at the blink of an eye, just like a lightning spark, okay? If it's too slow, then not all of your sarcomeres will contract at the same time, and then you end up with a muscle that doesn't contract correctly. Just think about how fast your eye movements are. Every time your eye is doing movement, okay, that is a muscle contraction, okay? So that's how fast those muscles are moving, and that's how fast those electronic signals are being moving and being transferred from your nervous system to your muscular system. Now, what happens that once your task is completed, there needs to be a way for one to turn off the signal too. It's kind of like you don't want your CEO to overwork all your employees and work until they can't go home. You gotta have lunch break. You gotta have, snack break in between, and then you can go home and take a break and rest. Same thing here. Your muscle fibers needs to rest. They can't remain contracted forever. You turn on the signal by releasing acetylcholine for the contraction. Now you need to turn off the signal so that you can get rid of acetylcholine to tell it to stop contracting. Okay, give your muscle fibers a break. It needs to go through that cycle of stimulus, contraction, and then relaxation. So how do we turn off the signal? How do we get rid of acetylcholine? This is what those moon-shaped things are for, okay? There are two ways that acetylcholine can disappear from the synaptic cleft. One way is that they can diffuse away, okay, just by diffusing, okay? Or there is an enzyme called acetylcholine esterase, which is what here was talking about. Acetylcholine esterase, short for the abbreviation for that is ACHE, okay? So those moon-shaped things are the ACHE, okay? Acetylcholine esterase, and that particular enzyme is for breakdown of ACH so that it no longer is active within the synaptic cleft and there's no more action potential coming at the motor neuron, because the whole thing that started the action potential on the muscle side is the fact that you have acetylcholine in the synaptic cleft. So if you get rid of them, then there's no signal anymore, okay? No ACH, okay? And then there won't be any more ACH release because action potential stops at the neuron side too. and the signal relay from the neuron to the muscle fibers will stop, okay? And here, then, that's when your muscle fiber is able to rest and relax and go through the relaxation stage of the muscle contraction cycle, okay? Now, this is not the end of the story yet. We have part 2 now, okay? So what needs to happen to the action potential that was relayed to the sarcolemma? We were just talking about the synaptic cleft. That's all we got done talking about. This next part, okay, is called the excitation contraction coupling, okay? What do I mean by that? Okay, so excitation part of that whole word, that part is for the neurostimulation followed by acetylcholine release into the synaptic cleft, producing an action potential across the sarcolemma in the muscle fiber. Okay, the action potential in the sarcolemma then will travel down. So this is that sarcolemma right here. That action potential will travel down towards what we call our T-tubule. Remember, we talked about these tubes, those yellow tubes wrapping around those myofibrils. Well, here is their role, okay? T-tubule is going to relay the signal of that action potential from the sarcolemma on the top, okay, towards the middle of the myofibril, okay, and then signal the terminal cisternae of the sarcoplasmic reticulum, which will then trigger the release of calcium ions from the terminal cisternae of the sarcoplasmic reticulum into the sarcoplasm. Okay, so this is what I'm talking about here. Okay, so again, you have neuron, axon, terminal, ACH release into synaptic cleft, activating action potential of the sarcolemma, which then action potential travels down the T-tubule. From the T-tubule, that's what's going to activate this part, which is the terminal cisternae of the sarcoplasmic reticulum, releasing calcium ions to the sarcoplasm, okay? And then, of course, calcium ions is going to go ahead and bind to your troponin, which I'm going to talk about next, okay? So calcium ion, after it's released from the sarcolemma, is then going to bind to that specific binding site we talked about on the troponin. Okay, troponin then rolls away the tropomyosin to reveal the active sites on G-actin of the thin filament, and this allows the myosin head on the thick filament to bind to G-actin. So this bondage between the myosin head and the actin is called the cross-bridge formation. Okay, so cross-bridge formation, that action of the myosin head binding to the G-actins. That's what this is illustrating here. What's happening is that when calcium ions binds to that little brown thing, okay, and that little brown thing is the troponin, and the green thing is the little green dot there is the tropomyosin. And when calcium binds to troponin, it rolls away the green dot, which is tropomyosin, reveal this yellow section of the G-actin. Then your myosin head, which is this guy right here, can then bind to that yellow region, which then this binding right here is what we call the cross-bridge formation, okay? Once cross-bridge is formed, the myosin head then swings in that one direction I was talking about earlier and pulls the actin towards the M line of the sarcomere, resulting in sarcomere shortening that results in a muscle contraction. Okay, so this is the whole muscle fiber, okay, and then every one of those little sarcomeres within each of those little myofibrils we're talking about is going to contract at the same time due to this whole process of excitation contraction coupling, which many of these muscle fibers will then contract at the same time to give you the whole muscle as a chunk to contract together to generate some sort of tension that's going to be able to power you to either lift something up or move your arm, for example. Okay. All right, so let's zoom in and take a look at that cross bridge formation and the swinging of the myosin head, that we actually call it the power stroke in detail. Okay, so this is about cross bridge formation and the power stroke of the myosin head towards the M line. Okay, so again, calcium ions were released from the terminal cisternae of the sarcoplasmic reticulum into the sarcoplasm. So here's that calcium ion right there floating around in the sarcoplasm. Now, calcium ions then will be able to bind to troponin. Okay, so there's that calcium ion binding to the brown troponin. Troponin, which then weakens the bond between actin and troponin-tropomyosin complex. Okay, troponin changes its position, rolling tropomyosin, this green thing, away from the active sites on G-actin. Okay, so you see this yellow dot right here? That yellow was not present here because the green line is blocking all along those active sites, along each of those G-actin. When it's rolled away, you reveal all those active sites on the yellow spots on each of those red spheres of G-actin. When you review those away, then you can allow the myosin head to bind. So active sites on the G-actin red spheres are exposed, which allows for your myosin head here to bind and form a cross bridge. Okay, so that's what we mean by cross bridge formation. The myosin head binding to the active site on the yellow circle. Okay, the yellow active site on the red ball. Okay, then myosin head automatically pivots, okay, and pivots towards the M line this way. Okay, and this is what we call power stroke. Okay, power stroke. And then ADP and phosphate, which was attached to the myosin head all along. You see how this myosin head already have an ADP and inorganic phosphate attached. Okay, so all along it's going to be attached. But once power stroke is done, this ADP and phosphate will be released from the myosin head. And now another molecule of ATP is now needed to release the myosin head from the G-actin and swing it back into the original position. Okay, so ATP binds, release that bondage of cross bridge, and then your myosin head will then swing backwards and sits back down in that resting position, and then it's going to get ready for that next binding, pull, sits back down. Next binding, pull, sits back down. Okay, you're gonna see what I mean when we go through that animation. Okay. All right, so notice that throughout this whole cross bridge and power stroke, an ATP molecule is not necessary to form the cross bridge between myosin and actin, and it is also not necessary for the power stroke to happen. The only place within cross bridge and this whole thing where ATP is needed is to release myosin head and return it to its original position where ATP is needed. Okay, so that's the only spot where ATP is hydrolyzed, is for that release of the myosin head from the actin and swinging back to its original position. That's when the ATP is hydrolyzed into ADP and its inorganic phosphate, and then now it just goes back to step one again and do the whole thing all over again. Okay, it goes through this many cycle of this binding to actin, pivot, binding to actin, pivot, binding to actin, pivot. And then that's what caused the whole actin filament of the sarcomere to move inwards toward the M line, and the entire sarcomere moves in, which then the whole myofibril contracts and the whole muscle fiber contracts. Okay? All right. So that's how the myosin head is able to be reactivated, is by hydrolyzing the molecule of ATP. Now, I know it's hard to imagine all of this happening with these images. So here's an animation video that goes through the whole process, the excitation contraction coupling between neuron and the muscle fiber, and all that needs to happen with the muscle fibers, thin filament and thick filament. Okay, so pay attention to this video, and it does an excellent job of going through. First, it goes through what's happening with the calcium ion and the troponin and tropomyosin, and then it goes back to talk about how the neuron releases ACH, acetylcholine, into the muscle fiber. Okay?
You use muscles.
Release that.
Every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay.
You use...
Oh, we don't need to replay that again. All right, so let's continue, and I want to make sure we're still recording. Okay. Now, a muscle fiber cannot remain contracted forever. Like I was saying, you need to give it a break. You need to let it relax. So, or else all your muscles will be frozen in place and the muscles don't know when to relax. So, muscle fibers must go through cycles of contraction and relaxation. And hence, there are a few things that control how long a contraction lasts. First, it depends on the period of stimulation at the MNMJ. Okay, so if action potential keeps coming, acetylcholine will continue to be released into the synaptic cleft. Action potential is going to be relayed to the muscle fiber. Okay, so that's what that's what regulates how long the contraction will remain. Presence of free calcium ion in the cytosol itself also has a role. Okay, so calcium ions remaining in the cytoplasm or sarcoplasm of the muscle fiber is what then allows the calcium ion to bind to the troponin. Therefore, all those steps happens with cross bridge formation and power stroke to result in a muscle fiber contraction. So, and the last thing is availability of ATP. If you don't have ATP, myosin heads can't release from the actin, and it cannot do another round of cross-bridge formation and power stroke. Okay, in order for it to continuously do that multiple times to cause the sarcomeres to contract, you have to have ATP around. Without ATP, everything stays frozen in place. Now, contraction will continue if there's more action potentials arriving at the NMJ in rapid succession. Okay, so that's what will continuously cause the muscle fiber to contract. As long as there's a signal coming from the neuron, your muscle will continue to contract. Now, so this is why acetylcholine esterase is so important in the synaptic cleft at the NMJ. It breaks the acetylcholine neurotransmitter, so it stops the action potential from coming at the sarcolemma on the muscle fiber itself. Okay, so no action potential at the sarcolemma, no action potential at the T-tubule, and no stimulation of sarcoplasmic reticulum to release calcium. Sarcoplasmic reticulum, once it sees, there's no electrical signal coming from the T-tubule, well, time to, gather all my stuff and call it a day. Okay, so sarcoplasmic reticulum will pump calcium ions back into the sarcoplasmic reticulum, and the concentration of calcium ions will then fall and detach from your troponin. Troponin then returns back to its original position. Tropomyosin then covers your active site back on the actin. Myosin head no longer can bind to those active sites. Contraction cycle then ends. Okay. So relaxation, in a sense, starts from acetylcholine esterase doing its job at the synaptic cleft in order to trigger all of this stuff that happens to tell the muscle to relax. Okay? So now that we understand the exact step where ATP is necessary during excitation contraction coupling, we can apply that to the idea of rigor mortis. Okay, this is interesting. I love this part. Okay, so when death occurs, circulations will stop, which means your blood stops traveling. Your skeletal muscles are deprived of oxygen and nutrients. Okay, and within a few hours, skeletal muscle fibers will run out of ATP. Okay, and without ATP, sarcoplasmic reticulum cannot pump calcium out of the cytosol. Okay, so that's two important function of ATP in terms of this whole muscle contraction relaxation cycle. Okay, ATP is critical for the cross bridge formation to be released. Okay, where the myosin head going back to its original position is where ATP is needed. The other spot where ATP is needed is that sarcoplasmic reticulum actually have to pump the calcium ions back in because, there's lots of calcium ions in there. So it can be going against its gradient. Okay, so that's why it needs to be pumped back, which then requires ATP in order to do active transport of the calcium ions back into the sarcoplasmic reticulum. Okay, so that continuous presence of calcium ion in the sarcoplasm is either via diffusion from the extracellular fluid. Okay, once the sarcoplasmic will, because the calcium ions is what trigger the contraction of muscle fiber. Okay, so when a person dies, everything stops. Okay, so there's no ATP. So that means ATP won't be there, and your sarcoplasmic reticulum can't pump the calcium ions back into the into itself. If it can't pump, that means all those calcium ions just remain in the sarcoplasm of the muscle fiber. Either they remain there, or things actually will start diffusing from the extracellular fluid space into the muscle fiber itself too. Regardless, after a person dies, calcium ions are still hanging around there where the muscle fibers are, because now there is no ATP to pump it back. So troponin is bound to the calcium. It remains bound because there's nothing to pump it back. And the tropomyosin will stay in that open conformation, exposing the active site on G-actin, which means your cross bridge is stuck there. You cannot release because there's no ATP, and all the active sites are open anyway, so they can form that cross bridge. In addition, because there is no more ATP around, the cross bridge between myosin head and actin actually gets locked into the position because it cannot release without a molecule of ATP. Your myosin head is stuck to the actin. So this is why we end up with the idea of rigor mortis after death. Okay, and rigor mortis is when the entire body's skeletal muscles become stiff. And this can last, 2 to 7 hours after death. That's why once a person dies, it kind of freezes into that position, is because your skeletal muscle fibers cannot relax due to inability to have ATP around. So that's why sometimes you can tell what the person is holding at the time of death due to their rigor mortis. Now, rigor mortis will go away, though. The rigor mortis will last until lysosomal enzymes comes in there to your muscle fibers and start breaking down those Z lines. Once a person dies, everything starts degenerating. Everything starts deteriorating, including your muscle fibers. So after a few hours, those enzymes can break down and chew up all those Z discs. And the whole sarcomere organization we're talking about with A-band, I-band, titin, all that stuff to keep it together, all of those falls apart. Okay, and when those all fall apart, that's when rigor mortis will then be relaxed. It doesn't matter that actin is still attached to myosin. The whole sarcomere is falling apart, and the whole muscle fiber is now falling into pieces. So that's when rigor mortis will go away. The body softens, and now you can move the body into that soft position. Okay, so that usually happens anywhere from 1 to 6 days after death. Okay, for rigor mortis to stop. Now, interestingly, some pesticide actually contains ACHE inhibitor, acetylcholinesterase inhibitor. Remember that we just talked about the importance of acetylcholinesterase is for the purpose of relaxing the muscle. So if you inhibit that enzyme cannot get rid of acetylcholine. Acetylcholine remains in the synaptic cleft, and you have that same idea of freezing your muscles in place, okay? And that's why when you are spraying that little pesticide, that bottle of pesticides on a little bug, the ACHE inhibitors within the pesticides is what is going to stop the relaxation cycle from happening in the muscle fibers of the bug, of the insect, okay? So then acetylcholine will be there in the synaptic cleft. They won't be broken down, and it results the muscles from not being able to relax, causing rigid paralysis. So that's why when you spray those pesticides on a bug, you kind of see them freeze in a little place, and the leg may twitch a little bit, and that's because of that inhibitor doing its job, freezing the muscles and paralyzing it from movement. Okay? All right. Okay, so that is usually the toughest part of the lecture in this chapter. So we're done with all this biochemical stuff now, okay? Now we switch gears to more physiology as a at the macroscopic level, part of muscle contraction. I talked about how a muscle fiber must go through cycles of being stimulated, followed by contraction phase, and then relaxation. So, a single stimulus contraction relaxation sequence in a muscle fiber is known as a twitch. Okay, one twitch is where the muscle goes through stimulus, contraction, and then comes back down in relaxation mode. And the twitch variation, how long a twitch lasts in a muscle fiber, can differ depending on the type of muscle. For example, eye muscle fiber twitch can be only 7.5 milliseconds. Okay, so this is what I was talking about, how fast those electronic signal can be and how fast those action potentials can go down the entire muscle fiber when I was talking about your eye movement. Okay, now, on contrast, you have your soleus muscle fiber, which is at the side of your leg, okay, side of your lower leg. Those muscle fiber, one twitch can last almost 100 milliseconds, even though it sounds like, 100 milliseconds, but that's still pretty fast. We're talking about millisecond, less than a second, which means just the blink of an eye, even less than that. All that electronic signal, all that cross-bridge formation we're talking about happens just like that, okay? That's how fast your body and your electrical nervous system and muscular system is working together for you to move, okay? Very quickly. Just imagine how quickly you know to swerve to the left if someone is cutting into your lane while you're driving. That's how fast everything is happening inside your nervous system, okay? All right. So a myogram is a graphical representation that shows tension development in muscle fibers. You have different phases within. So this, for example, is a twitch of an eye muscle. This is a twitch of your gastrocnemius. This is your twitch of your soleus. And they all differ in time. But what is this curve representing? What are the different phases? This is what the myogram does. Okay, so this is a myogram showing you a stimulus and then a contraction phase, and then followed by a relaxation phase. So we're going to talk about these phases and periods a little bit. So latent period right here is the period of waiting for that stimulation to come through, okay? Action potential needs to sweeps across the entire sarcolemma for your sarcoplasmic reticulum to release calcium ions. So that's that whole latent period of waiting for calcium ions to get to every one of those troponin in every one of those sarcomeres. And then the contraction cycle right here is the contraction phase where your muscle gets, your myosin are pulling the actin in one by one, section by section, until your tension of the muscle fiber increases to a peak, okay? And that is where your maximum tension is happening. Calcium ions is now bound to troponin. Active sites on thin filament is exposed, and then you have those cross-bridge formation. And then after contraction phase, you have the relaxation phase. Relaxation phase is where the calcium ion will decrease because they're pumped back into the sarcoplasmic reticulum. Active sites are now covered again by your tropomyosin, and the number of active cross bridges will decline, allowing your muscles to relax back to its original resting length. Okay, so now that what we have been talking about is just one single cycle of contraction, a twitch. But to lift something up, for example, if you were to pick up a coffee mug from your table right now and hold it in the air, that's many of your muscle fiber during many cycles of contractions. And depending on the activity, one may need a bit or a lot of contraction depending on what you're trying to do, how much tension you need to generate for that action, and you may need a lot or a little bit of contraction, okay? They will produce different amount of tension power. So, for example, if a muscle fiber is just warming up, not really, needed to lift anything per se, but the stimulus comes, contraction happens, and then relaxation happens. But it doesn't end in one twitch cycle. This is what we're looking at here, okay? It goes through contraction, relaxation, and then immediately a second stimulus comes and arrives, and the muscle fiber starts contracting again. This warm-up is called a trep. What happens is that there's too little time when the relaxation of the muscle fiber starts to happen to allow to all the calcium ions to be pumped back into the sarcoplasmic reticulum. Because if we think about it, if we're just talking about twitch, it goes through contraction, relaxation, and then a little bit, and then latent phase once more time, and then contraction, relaxation. What happens in a trap is that you're not allowing this whole relaxation and latent phase to begin again. You're immediately hit with the second stimulus to come through. There's no time for the sarcoplasmic reticulum to pump all those calcium ions back in, so calcium ions are remaining within the cytosol. So that's why then you end up with a little higher, a little higher, a little higher, a little higher of that twitch pattern throughout the entire muscle contractions, okay? More than one contraction going on, okay? Now, if we need to lift something and actually want to result in a more powerful overall muscle power, because you can see this graph here on the y-axis represents the tension power. So a trap doesn't really generate that much of a tension power, okay? We can't lift anything with that. So there needs to be something that produces a more powerful overall muscle tension. So this requires something called wave summation. What happens here is that you have your first stimulus here on your first cycle, muscle fiber contracts. But before it can even relax fully, a second stimulus comes in for signaling contraction. So it ends up being added to the tension power of the two twitches. And this can go on and on and on to produce an overall more powerful tension that allows up to power a movement. Okay, so for example, you have here stimulus, contraction. Before it can fully relax, another stimulus comes through, contraction, and then before it can relax, stimulus comes through again, relax a little bit, and then another stimulus comes through. So it keeps adding up the tension power each time that stimulus keeps coming very, very quickly through. So you're adding that tension power of these twitches together to give you a more powerful movement. Okay, so let's say we're just lifting a pen from our desk. Well, it really doesn't require a lot of muscle power to lift a pencil from your desk. So we don't need to utilize that full potential tension power of our muscles to lift that pencil. We're not struggling to lift that little pencil up, okay? We are just, lifting it up, okay? But we still need to induce contraction in form of that wave summation in order to generate enough tension, just enough to lift that pencil so that it is called an incomplete tetanus. Incomplete tetanus is generating that overall muscle power, enough tension to lift whatever you're doing. You're not struggling, you're not maximizing the power of your muscle, or else you will end up swinging that pencil to your face. You don't want to do that. So you just kind of, pick up my pencil and do my thing, okay? That's incomplete tetanus. You're not using or, maximizing that power within your muscle. Now, let's say instead of lifting your pencil, we want to lift up our printed anatomy textbook from our desk and hold it above our head using one arm. Now, that is a heavy book. Okay? All of you are probably looking at digital versions of your book, but I have printed version of the book that is pretty heavy, and you may need to activate the full capacity of tension power, your muscle fibers in your muscles, in order to sustain that weight of the book above your head. You can't go any higher because you have reached that maximum tension power. Now, I'm not talking about you men with really big muscles that you can just lift that anatomy textbook. I'm just talking about, imagine, a skinny female, okay, with not a lot of muscles. You don't work out, okay? You don't usually go out to run or jog or do your weightlifting, okay? So you're doing your best trying to lift this book with one hand. Oh, and eventually, at first you feel pretty okay. I'm doing fine. And, you hold it there for 5 minutes and you start feeling very tired. So because you're generating and reach that maximum tension power in your muscle, eventually your muscle fatigues, feels tired, and you are forced to put the textbook back down on the table because now your arm is all tired because the muscle fibers weren't allowed to relax at all. You see, here at least they're able to relax, contract a little bit, and keep holding that pencil. Now you're really maximizing that maximum power. It's remaining in contraction all that time. No relaxation at all. That's why then your muscle, I feel tired. I got to get back down here. So that's when you put the book down and feel, finally, now the textbook is back on the table. This is what we call a complete tetanus, is when your muscle has used its maximum potential power. So I guess for people that are more built, we're talking about maybe a 50-pound bag of rice with one hand, okay, versus a textbook, okay? Everybody's different. But when your muscle reach that maximum maximum power tension that your muscle can generate, that's what we mean by complete tetanus versus incomplete tetanus, okay? So lifting something very heavy for complete tetanus and lifting something light like a pencil for incomplete tetanus. And of course, the whole lifting of the pencil in the textbook story doesn't only involve one muscle fiber, but lots of them working together to produce that overall tension power. A typical skeletal muscle can contain thousands of these muscle fibers. Now, we talked about how one muscle fiber can only be innervated by one motor neuron, but one motor neuron can actually control multiple fibers, okay? So multiple axon terminals can go one. For example, you see this purple neuron here going from the spinal cord to your muscle fiber. One axon terminal goes here, another axon terminal goes here, another axon terminal there, goes there, okay? So all of these purple muscle fiber are innervated by that one purple neuron, okay? Same thing for the blue neuron. The blue neuron has these, different axon terminal. And then these axon terminal can be sitting on different muscle fiber, all the blue ones here, and then the red neuron is controlling all the red ones here, okay? And hence, one motor neuron along with the multiple muscle fiber that it controls innervates is known as a motor unit. Now, why this is the case is that imagine we talked about how there's thousands of these muscle fiber within your muscle. So if there was only one neuron for one muscle fiber, you would probably have too many neurons going on. You need thousands of those motor neurons for each of those thousands of muscle fiber. That's not efficient in, saving space. Our body is already compact enough with all that little stuff in there, blood vessels, lymphatics, everything, muscles, organs, etc. So we need to save space. So this is why we have one neuron controlling multiple muscle fiber at the same time. Okay, and hence what we have with the motor unit is that the bigger a motor unit is, meaning one neuron controlling lots of muscle fiber, the more rough the movement. Okay, the bigger the motor unit is, the more rough the movement. We're talking about swinging your arm up in the air, okay, in this big movement, okay? A smaller motor unit is where one motor neuron may only control a few muscle fibers. So the smaller a motor unit is, the more precise the movement is, okay? So for example, a large motor unit would be utilized to lift the anatomy textbook, okay, up in the air. But a small motor unit would be used instead to do a fine motor skill like sewing, this little crafting. Okay? If any of you do that Swarovski sticking on your phone or something, that's fine motor skills. So those would require a smaller motor unit versus you picking up your backpack and walking out the door. That picking up your backpack requires a larger motor unit for that whole rough muscle moving. Okay? All right. The other thing that we have to be aware of is that even when a muscle is not necessarily contracting, some motor unit is always active. This resting tension is what we know as muscle tone. So different motor units are always stimulated at different times so individual muscle fibers can relax. So even though you're just sitting there not really doing anything, the fact that you have muscle tone means that your muscle motor units are taking turns doing something. They might not be at complete tetanus, okay, but they may be doing something. They were doing some contraction of some sort, even when we're just sitting down. So, activated muscle fibers will use energy. Okay, so the greater the muscle tone, the higher the resting rate of metabolism there will be. Okay, so when you have a bigger piece of muscle, it will require more energy, even when you're just resting. Of course, when you're activating it to do stuff, lift weights, go out and have a run, a jog, then that really requires and burn a lot of ATPs. But when you're resting, those larger muscles will also still burn energy because of this muscle tone we're talking about. So this is why they say one of the things about losing calories and losing weight is to build muscles, because even when resting, having larger muscles will always burn more energy to keep the muscle tone going, okay, compared to small muscles. And in terms of the overall muscle contraction, there are different terminologies related to the length of the muscles at varying degrees of tension. So isotonic contraction refers to when the contraction or the tension generated by the muscle increases and the length of the muscle will change in response, depending on how much tension that is generated or that is needed for pulling up a weight, for example. So let's say if the muscle tension is able to exceed the load that it needs to lift, and the muscle will then shorten in contraction. So this is a 2 kilogram load. You're like, okay, that's light. Okay, I'm going to pick it up, and now I can pick it up in the air with my arm. So your arm muscle has shortened to be able to pick up that weight. So this is called isotonic concentric contraction. Isotonic meaning your tension increases. Concentric means that your muscle actually shortens in response. Now, if the peak tension ends up not being able to lift the heavier load and the muscle elongates instead, this is what we call isotonic eccentric contraction. So let's say now instead of lifting a 2 kilogram weight, we're lifting a 6 kilogram weight. And then let's say that I can't lift 6 kilograms. Well, I'm struggling. I'm already at maximum tension and I can't lift that up. So now my arm remains extended down and I can't lift that weight out. Okay? So your muscle at that stance right here, okay, is in that isotonic because you are generating tension, but you are already at your maximum tension, not enough to pull and shorten that weight. So that's why it's isotonic still, but it's eccentric now because now your muscle cannot shorten, but it lengthens instead because the power you generate is not enough to pull up that load. Okay, and the load is pulled down by gravity instead. All right, so now if the muscle tensions generated is equivalent to the load and muscle does not change in length, as a result, this is called isometric contraction. So isotonic is generating tension that can vary the length of your muscle. Either you're shortening it in concentric or you're lengthening it in eccentric. But if you're neither shortening or lengthening, it's called isometric contraction. Let's say you're strong enough to hold that weight just in place. You're not going to move it up in the air or you're not going to extend it downwards. You're just going to have it stay in place in front of you, okay? Holding that weight, withstanding that weight. That doesn't mean you're not generating tension. You're still generating tension just to hold that weight in place. But that tension does not exceed the load, so you don't shorten your muscle. And that tension is not low enough that you actually end up lengthening your muscles, okay? So This is how, in a sense, we are working out our muscles by varying our degree of muscle tension generated when we're lifting and putting down a dumbbell like this when we work out. So this is what I'm talking about. Let's say now we're not changing our weight like I was talking about. We're all trying to lift up this 5-pound dumbbell, okay? So when we're doing that flexion movement, moving that weight here and then putting it back down, we're actually using our nervous system to tell our muscles how much tension to generate that results in pulling your dumbbell upwards or putting the dumbbell down. You're varying that tension that is generated, okay? So when you're putting that dumbbell like this, holding it upwards, not really moving it anywhere, your muscle does not shorten. This is called isometric contraction. You're just holding that dumbbell in place. Now you want to move up that dumbbell towards your biceps. Okay, so you're doing a flexion movement at the elbow, and that is resulting in a shortening of your muscle. That's why you can do the flexion movement, and this is what we call as isotonic concentric contraction. Now, you have to put dumbbell back up so you can continue doing cycles and rounds of this moving up your dumbbell. So now you're going to extend your muscle and allow the dumbbell to come back downwards by gravity, okay? And that is that extension movement you're doing, and you're lengthening your muscles, which means this part of it is called isotonic eccentric contraction. So you're going through cycles of isometric, isotonic contraction, concentric contraction, isotonic eccentric contraction. You're going in cycles of these when you're lifting and putting down that dumbbell in cycle while you're working out your muscles at the gym, for example. Okay? All right. So it's been an hour, so I'm going to stop part 2 here and save the rest for the last part of this chapter. Let's take a break.